Welcome to this lecture on genomics in computational biology. Today, we are going to explore how scientists read and understand the massive instruction manual that builds and maintains every living organism. It is a fascinating journey at the intersection of biology and computer science. And by the end of this session, you will understand exactly how we piece together the code of life. Let us start with a simple analogy. Imagine you are trying to build a massive, incredibly complex Lego castle, but the instruction manual for this castle is written in a language with only 4 letters: A, C, T, and G. In biology, this instruction manual is called a genome. Every single cell in your body carries a complete copy of this manual, tightly packed inside its nucleus. Genomics is essentially the science of reading, translating, and understanding this massive instruction book. Instead of just looking at one single Lego piece, genomics looks at the entire castle and how all the instructions work together. Now that we have an intuitive grasp, let us look at the formal definition. In scientific terms, genomics is an interdisciplinary field of biology, focusing on the structure, function, evolution, mapping, and editing of genomes. A genome is an organism's complete set of DNA. This includes all of its genes, as well as the non-coding sequences that regulate how those genes are expressed. While traditional genetics might focus on studying individual genes and their specific roles in inheritance, genomics takes a holistic, system-wide approach to analyze the entire genetic material simultaneously. Understanding this holistic approach brings us to a major challenge: the sheer scale of the data. Genomes are incredibly large. For instance, the human genome contains over 3 billion base pairs. Because of this massive size, scientists simply cannot analyze them manually with a pen and paper. Therefore, genomics relies heavily on computational biology and bioinformatics. We must use powerful computers, advanced algorithms, and sophisticated statistical models to process, store, and interpret these massive amounts of DNA sequence data effectively. Without computation, modern genomics would simply not exist. So, why do we go through all this computational effort? Understanding genomics is absolutely crucial because it holds the key to unlocking the mysteries of human health, disease, and evolution. In the real world, genomics is the foundation of personalized medicine. This is where medical treatments are tailored specifically to your unique genetic makeup. For example, if a patient has cancer, doctors can sequence the genome of the tumor to figure out exactly which genetic mutations are causing it, allowing for highly effective targeted therapy. Beyond human health, it ensures global food security by helping engineer crops that survive droughts or resist pests. This incredible real-world impact leads us to how we actually read the DNA. When we sequence a genome, we cannot read the whole thing from start to finish in one continuous go. The technology does not allow it. Instead, we chop the DNA into millions of tiny fragments, read those individual fragments, and then use computers to stitch them back together like a giant jigsaw puzzle. To ensure we have read every part of the genome accurately and haven't missed critical pieces, we need a way to measure our success. We need to know how many times, on average, a single letter in the genome has been read. To measure how well we have read the genome, we use the fundamental Lander-Waterman equation. Conceptually, this formula takes the total amount of DNA data we generated and divides it by the actual size of the genome. Let us look at the symbols. C represents the average sequencing coverage, which is a dimensionless number telling us how many times a base is read. L is the length of each short DNA fragment in base pairs. N is the total number of these short reads generated by the machine. Finally, G is the total length of the target genome in base pairs. If the number of reads, N, increases, the coverage C increases, meaning we have a more reliable and accurate genome assembly. Now that we have our formula, let us see this with a concrete example. Imagine you are tasked with sequencing a newly discovered bacteria. We are given that the total genome size is 4 million base pairs. Our sequencing machine generates 200,000 short reads, and each read is exactly 150 base pairs long. We need to find the average sequencing coverage. We choose the Lander-Waterman formula for this. First, we multiply the read length of 150 by the 200,000 reads to find the total amount of DNA sequenced, which is 30 million base pairs. Then we divide this by the genome size of 4 million. The final answer is 7.5. This means every single letter has been read 7.5 times on average. So we have calculated an average coverage of 7.5. This brings us to an important question. Is average coverage enough? Because chopping up DNA for sequencing is a completely random process, the coverage is not perfectly even. Some parts of the genome might be read 20 times, while other parts might, purely by chance, not be read at all. This is a huge problem if the missing part contains a crucial gene mutation. Therefore, we cannot just rely on the average. We must mathematically determine the probability that a specific base in the genome is completely missed, meaning it has zero coverage. To find that probability, we use a concept from statistics known as the Poisson distribution, specifically the formula for zero occurrences. Conceptually, this models the chance of an event not happening at all in a random distribution. In this equation, P of 0 represents the probability that a specific DNA base is sequenced exactly 0 times, meaning it is missing from our final data. The symbol E is Euler's number, a mathematical constant approximately equal to 2.718. The symbol C is the average coverage we calculated previously. Notice the negative exponent. This tells us that as our average coverage C increases, the probability of missing a base decreases exponentially. This is why scientists always aim for high coverage, like 30 or 40. Let us apply this formula to our previous bacterial genome example. We are given our previously calculated average coverage of 7.5. We are asked to find the probability that a specific crucial gene mutation is completely missed because it was sequenced 0 times. Because the fragments are generated randomly, we choose the Poisson distribution formula. We substitute our coverage value of 7.5 into the negative exponent of Euler's number. Calculating e to the power of negative 7.5 gives us approximately 0.00055. Physically, this means there is a 0.055% chance that any specific base is missed. In a 4 million base genome, about 2,200 bases would be completely missing. Now that we have calculated these values, let us visualize what sequencing coverage actually looks like. In this diagram, the long, continuous line at the very bottom represents the full reference genome. Above it, you see many short, overlapping horizontal lines. These represent the millions of short DNA reads generated by the sequencing machine. Notice how they are scattered randomly. The vertical dashed box highlights one specific location on the genome. If you count the short reads inside that box, you will see exactly 5 fragments overlapping that specific spot. This visually represents a coverage of 5x for that particular base. It perfectly illustrates why we need high average coverage to ensure no gaps are left in our final puzzle. We have covered a lot of ground today, so let us summarize the key takeaways. First, genomics is the comprehensive study of an organism's entire genetic blueprint, and it absolutely requires powerful computational tools to piece together massive amounts of DNA data. Second, we learned that genomes are sequenced by breaking them into smaller, readable fragments. Third, we used the Lander-Waterman equation to calculate average sequencing coverage, ensuring we collect enough data. Finally, we applied the Poisson distribution to understand the probability of missing data due to randomness. Mastering these foundational concepts is your first step toward turning billions of raw DNA letters into meaningful, life-saving biological insights. Great job today!